Okay, and now we go to our last speaker, who is Paul Stys from Sorbonne University in Paris.
Hello, sorry. Hello, I'm a PhD student in the history lab. The immune system is a dynamical biological object whose modification will depend on cell population and the amplitude of the response according to the type of antigen encountered, microbial or self-antigen. It can be assumed, as shown in some papers, that this dynamic results from an activation of specific T-cell receptor. The TCR could be used as a biomarker of a pathology. Our objective are to identify disease-specific TCR from the repertoire of sorted T-cells, use these sequences to classify and predict patient outcome, and determine the specificity and biological roles of this TCR to better understand this disease. We launched two observational trials. The first one is Transimmunome to revisit the nosology of autoimmune diseases. At this time, we focus on type 1 diabetes and rheumatoid arthritis. These trials also include healthy volunteers. And the second trial is Sirocco, aimed to investigate the mechanism of a cell subset in COVID-19 patients. For each trial, we sort CD4 T-cell subpopulation from peripheral blood, effector T-cells and regulatory T-cells, and second, their TCR alpha beta repertoire in bulk. We focus the analysis on CDR3 amino acid. After the application of our identification strategy, which I will not detail here, we isolate for each disease two signatures. One that corresponds to the anxious sequences in disease, and one corresponds to the improved sequences. A very important point for us, beyond the identification strategy, is to validate the approach and answering the results. We use two approaches for that. The first one is to use the machine learning to train a model on the 75% of the patients with whom the signature was made, and then to classify the remaining 25%. The second approach is to use external dataset. After applying our strategy to the Transimmunome dataset for autoimmune disease in left and to the Sirocco dataset for COVID-19 in right, we identify disease-specific CDR3 amino acid. These sequences are represented here as a heat map with their presence in yellow and absence in black across all the patients. For more clarity, only the CD4 effector represent are shown here. One clustering method is applied on the patient to discriminate who shares the same sequences. For the signature identification in each disease, it is specified if the sequences is enriched in the disease or either impoverished, and if the sequences come from the alpha or beta chain. We see blocks of sequences specific to each disease. This is clear for COVID-19 and allow a better clustering of the patient. This selection of CDR3 amino acid from peripheral blood works well on the viral disease like COVID-19, since the clustering shows a perfect separation of a patient from healthy volunteer. The strategy also separates autoimmune disease patient from healthy volunteer, but to a lesser extent. A first validation is the comparison of our strategy versus a random signature. A random signature has the same size as the one identified, but with sequences drawn from the whole dataset. The red vis represents the CDR3 expression of the 4 autoimmune disease signature for each patient. The signature was calculated on 75% of patients of each disease. 6 random draws of the dataset were performed. It's the iteration. The random signature did not separate the patients, whatever the iteration, which shows that the strategy is reproductive, little impact by sampling, and the results are not due to chance. The second validation of it, in fact, I will be discover the rest by going to my poster, the number 2002, or on UVA. To conclude, I would like to thank my team I work with in E3 Lab and our collaboratory. Thanks for your attention.